Alright, howdy everyone, my name is Trisha, I'm from Insectopia, and today we are going to be talking about a variety of butterflies and moths. Alright, the lepidopterans are the butterflies and moths. Perfect. Alright, so we're going to be talking about the butterflies, moths, and the skippers. We'll be looking at what that means. The Hesperiids, they're their own family, but they kind of break all of the rules of both butterflies and moths. So they get their own subcategory. That's fine. Okay, so funny thing when we're going through and identifying butterflies and moths, a lot of times the characteristics are wing venations. Alright, so we're going to be looking very, very closely at the wing venations in a lot of these guys. I will tell you kind of the key characteristics that I close in on when I'm looking at specimens too, kind of things that I've noticed over, the little bit of time I've been collecting and stuff. So we'll be able to go over all of those. Now, if you don't know what the difference between a butterfly and a moth is, I wanted to go over that very quickly. Butterflies are going to be mostly day flying, alright? They tend to have narrower bodies than moths. Moths tend to be kind of big, heavy-bodied insects. Butterflies fly in the day, moths a lot of times fly in the night. And then a big characteristic are those antennae. So you can see right here, this is my monarch butterfly. I'm using a yellow colored pencil so you can see it. You can see right here, my monarch butterfly has knobbed antennae. So they're long and straight and they're expanded at the end. That's going to be a characteristic of butterflies. Whereas, if we look at, we'll be able to see this guy's moth antenna a little later, but if we look at this guy's antenna, they're large and feathery, right? So they have the ability to smell from long distances away, and it's the males of this family that have those huge feathery antennas. Perfect. So yeah, there's going to be differences in when they're flying, there's going to be differences in their body shape, and there's going to be differences in the antenna. You can see this is my butterfly's body, right? It's long and thin. And then if you look over here at my moth bodies, these guys are going to be kind of big and plump and very, very fluffy. I love moths, so we're going to be looking at a lot of moths today. Hi, Sewing Nancy. I hope you're doing well today. Awesome. So that's going to be the big difference between butterflies and moths. Skippers are going to have their own characteristics, and we can see, let me grab a skipper really quick for you. The skippers, a lot of times, have hooked antennae. So instead of having just an individual knob at the end of its antenna, the antennae actually have that, there we go. The antennae have that hook at the end, and that's characteristic for just skippers. They also hold their wings kind of funny. We'll talk about them when we get to them. Cool. Alright, so the first family that I wanted to talk about are the Nymphality. And the Nymphality, their common name are the swift moths. Now, with the Nymphality, a lot of times when we're, alright, I'm going to show you, FW stands for front wing, and HW stands for hind wing, just so that you know as I'm typing these out. So the front wing and the hind wing venation is very similar. A lot of times there's large differences between the front and the hind wing venation. So if we're looking here at my monarch, you can see that there's this large cell up here, and then there's some breaks. But then in the hind wing, there's kind of a smaller cell, and the breaks are in different places. The wings are different, even shaped differently, right? So the veins are going to be different. Whereas with our Nymphality or our swift moths, those are going to have very, very similar wing venation. That's fine. All right, so let's check that out. We have, pull it out. This is our Hepielid. This is our swift moth. They are not common they're not common in Michigan. This is where this one was collected. They're more common in other areas of the country. You'll notice that the front wing and the hind wing have oddly the same shape. If you took the hind wing and you broke it off and you put it on top of the front wing, it almost looks like it could be the same wing, right? And so we're going to put this guy's wings, we're going to put him under the microscope, and we're going to look really closely at the veins so that I can show you how similar they are. All right, so here we are. This is the front wing of our swift moth, of our Hepielid. And when I say wing venation, we're not talking about these colors, all right? Those colors are just the scales on the wings. Now, when I say wing venation, we're actually looking for kind of these rised lines in the wings. These veins were used when the moth or butterfly emerged, and they would have pumped their body fluid through these veins to help expand their wings. But then, eventually, they pump their wings completely dry. So these wings no longer have any hemolymph running through them. There's no insect blood in these wings. All right. So if we're looking at the wing venation, we see some veins here. There's a break up here. We've got one. We do have this interesting Y here. That's going to be one that you can notice on the hind wing. So there's this Y right here. All right. We'll take a picture of that, keep it in our heads, and then we're going to go and jump down to this hind wing. So now you'll notice the hind wing still has that upper vein right up here that kind of branches in a bunch of places. I don't think we saw all of them just because there's so much color up there. But then you can see that we've got this cell, and it even has this individual Y. All right. So this is going to be a swift moth. All right, and that's because the front wing and the hind wing venation is pretty much identical. That's going to be your deciding characteristic for this family. Hi, Bushra. How you doing? Checking out butterflies? My YouTube just went down. Give me a second. I think they can still see it. I think I'm still connected. Alright, I'm going to have to use this computer really quick. Alright, give me 2 seconds. My other computer just crashed. Sad day. Doop, 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 doop. Oh no. Alright, we're all back. Oh, thank you. I appreciate that. I spent a lot of time on the butterflies and moths. I will admit that there are I will admit that there are a handful of specimens that I pinned when I was pretty young, and so some of them aren't as perfect as I'd like them to be, but it's good to have old and new. Kind of like actually the next specimen that we're going to be looking at. The next specimen I collected a little bit ago, so it's going to be, let's see, we collected this one in 2008. So I actually collected this moth before I graduated high school, guys. So the moth is spread and pinned, except that I would have pulled the front wings up a little bit, you know, to make sure that that line went all the way across. But that is all right. Now, the next family that we are going to be talking about are the Pyralidae. And these are going to be called the snout moths. Sammy! Hey, Sammy! All right, I'm back. Okay. All right, so we're talking about the Pyralidae. These are the snout moths. The fun thing about the Pyralids is that the snout moths, the characteristic isn't really on the wing venation. The characteristic is on the head. They have expanded, what do we call them? I guess they're expanded labial palps. Yeah, so they are actually named after these snouts. We talked about palps a little bit when we were talking about beetles. Palps are mouth fingers, right? But for butterflies and moths, they don't really need anything to help grab and pull stuff to their mouths, right? So I'd like to kind of just go and check out the snout. Yeah, Sammy the kitty, I had to run after her. But she's fine. Sometimes I'll run away to help save a kitty. Okay. Oh right, we were doing this. All right. We're going to zoom in on the head of this moth because it is actually kind of funny looking. All right, so we're looking at the head of this moth. And so this is the head right here. You can see right about here, it's got a compound eye. This is the where the antennae are connected, right? These are its antennae. The proboscis would be down here, kind of all curled up. And then we've got these things. All right, these are the expanded labial palps, and it almost makes it look like he's got like this frontward facing snout. Or like a really fancy mustache, right? But those are actually, they're adapted mouth parts for this moth, okay? So sometimes they'll look like they're a bonus pair of antennae up there in the front because they're so big, and when they're alive, they generally face forward. And sometimes they'll bring them up like this, like maybe even a rhinoceros horn, right? But that's going to be the characteristic for the family. Pyralids or snout moths are the only family that have this characteristic. And so that makes it pretty easy to identify those guys. have our pyralids taken care of. The next family that I wanted to talk about was Hesperiidae. The Hesperiids are the skippers. All right. A skipper is neither a butterfly nor a moth. All right. It is its own thing. It has its own group of insects. And you'll see that it has those very, very hooked antennae. Oh, let me write these down so that we can be following along. Good girl. Okay. The Hesperiids, they're going to have those hooked antennae. And they are going to, in the wild, if you see them, they'll hold their wings. It almost looks like they're partially open. It's kind of like at a 45-degree angle. And so if you'll think, a lot of times moths, depending on the family, sometimes they'll lay with their wings completely open, right? Sometimes butterflies and moths will kind of flap their wings in between, kind of fully open and all the way closed, right? We got some butterflies that rest closed. And the Hesperiids, or the skippers, that when they land, they generally land with their wings like this. And sometimes their front wing and their hind wing are actually at different angles. So the front wings will kind of be like this, and then the hind wings will be just a little bit more open. And you can distinctly see all four wings a lot of times when you're identifying skippers. All right, let's check this guy out under the microscope. We might as well while we have it. So there is going to be our hooked wings, or that's going to be what our hooked antennae look like. All Hesperiids are going to have them. I'm not sure which species this one is. A lot of times when you're identifying them, they can be a little bit difficult because the veins in the wings can be very difficult to see, and then some people will actually take their wings and rub all the scales off of them to identify the butterfly or the moth. Except that once you've wiped all of the scales off of your butterfly or your moth, then it no longer has any color. And so that is not my favorite thing, because then you have a bunch of clear guys that used to be beautiful, right? Alright, so those are going to be our Hesperiids. And the next family that I wanted to talk about, we're going to get into some butterflies. I have 4 families of butterflies to show you today. And the first one is Papilionids, or they are called the Swallowtail butterflies. We're just going to call them the Swallowtails. We'll call them the Swallowtail butterflies. They'll go in the next line. That's fine. Alright, give myself a little bit of space. Perfect. Alright, so with the swallowtail butterflies, a lot of times they will have a tail. Alright, and so the swallowtail butterflies 90% of the time have a tail. There are some species of swallowtail butterflies that don't have a tail. So you gotta be a little bit careful with that one. The swallowtail butterfly that we have today, this guy right here, and he does have at least one tail, right? This is his tail, the swallowtail that they're known for. The other one, unfortunately, got knocked off at some point. These swallowtails do have a purpose. So when these butterflies are kind of flying through the air, it might be kind of hard for birds to tell which side's the head and which side is the back, right? And so if they come and they try to bite at the butterfly, sometimes they'll bite at the tails, right? And then the tails will break off and they're safe, right? The swallowtail butterflies, they don't need the tails to survive. They can fly around perfectly without them. They're mostly decorative, I guess, and to help them defend themselves against birds. Alrighty, so we've got those guys. Now, the other characteristic on swallowtail butterflies is a characteristic that's actually on the front leg. Swallowtail butterflies, you can imagine them as being very, very clean. They're clean butterflies. And so they have front legs with essentially an antennal cleaner on their leg. So let's see. We're going to call it an appendage. All right. And make sure that this is safely placed on my microscope before I... All right. It's too high. Alright, I actually have to move my microscope. I have to adjust my microscope. Give me 2 seconds. Silly microscope. That's alright, I don't need to look into the eyepieces. You know what's funny is I adjusted it too high. so let me see if I can get this all focused here. That actually took a minute for me to find this guy. Alright, so if we are looking, this right here is the first leg's femur. This right here is the femur. That means this second part right here, that is going to be the tibia, or the second part of the leg. And if we look right about here, there's going to be a larger area of kind of longer hairs. It looks almost like a brush area. There's a little place that essentially the swallowtail butterfly is going to run its antenna underneath this portion of hairs, or this area, and it'll be able to clean its antenna. Alrighty. 90% of the time, you'll actually be able to tell that it's a swallowtail because it has tails, right? But if it doesn't have tails, that's the characteristic that you're going to have to use. You're going to have to use kind of that antennal cleaner on the tibia of the front leg. The next family that we get to do are the Pierids. These guys are going to be the Sulphurs. All right. There's a number of common names for butterflies in this family. They can be sulphurs, they can be whites. And they're generally the first butterfly that you see every summer. The cabbage white is generally the first butterfly that pretty much everyone sees. If we're looking at two different types of Pierids, that would be these guys, right? So a lot of times Pierids are going to have these spots on their front wings. Not all the time, but they do a lot of times. Generally, they are medium-sized butterflies that are white or yellow. All right, they tend to flutter around all over the place. And their hind wing, if you look at the hind wing is, that's why I don't use my finger, right? The hind wing is nearly 100%, it's nearly circular, right? So you see how the hind wing kind of orbs out like that? That's something that we're going to be watching out for. These guys are not as clean as the swallowtails, all right? They're not going to have that antennal cleaner on the front leg. But they are going to have split toes. I'm checking the legs on these specimens to see if we'd be able to see them. This guy's legs are out. Maybe we'll be able to see them. He's all white, so I'm going to flip the background. Let's see. Good girl. Her toes are so tiny. All right, right about there might be the best I'm going to be able to get for her. Oh, come on. All right, we're not going to see this one. That one is just too small. Now, what I was going to show you are they call them bifurcated tarsi. So they have, essentially, at the ends of their legs, they have a split, like this. And so their tarsi are bifurcated, or they're split into two at the end. But it was kind of small to see. You guys don't need to see me fidgeting with the focus all night. Okay. Let's see. So let's write those period characteristics down. So the sulphurs, they have nearly circular hind wings. And they're going to have bifurcated tarsi. That just means that their tarsi split in two at the very end. Alright, the next one, these guys are fun, and honestly, they're called the blues, and they're blue. This family is going to be difficult to misidentify, I think, because a lot of them are small and delicate. All right, they're generally, if you've seen a small itty bitty, if you've seen a small itty bitty blue butterfly, you can call it a blue. You can also call it a hair streak if you'd like. These are the lysinids. All right. Now, their antenna, good. Sewing Nancy, you'll love the antenna on these butterflies, because the antenna are ringed black and white, so they kind of look all striped, and that's gonna be a lot. That's kind of cool to see under a microscope. The other thing that they have are a line of they have white scales around their eyes. Alright, so, if we want to check a little blue out, I only have one, but I am proud of the spreading job on this guy, because normally, when they're really small like this, I didn't get them done very well. Alright, perfect. So that's going to be our lysinid. It has faded a little bit since I collected it, so it's almost down to kind of this gray color. But I promise you, when I collected it, it was blue, and we'll look at it under the microscope. I think it'll show up a little more blue under the microscope. And then we'll be able to see the antenna and all the fun things. Oh no. I think I glued this antenna back on at some point in its life. I think that's a little droplet of glue. That's funny. All right, these are the antenna of our lycaenid, and you can see that they have those stripes along their antenna, right? They alternate black, white, all the way up. They're all stripy and pretty. That is definitely a characteristic of this family, the lycaenids. And then, if we zoom in even closer and check out its eyeball, let's see. This is up, this is down. All right, so if we check out his eyeball, you can see that it's dark and then it's automatically kind of this white around the compound eye. All of the lycaenids are going to have those white scales outlining their compound eye. So you can see right here, this is our compound eye. These are the striped antenna. And then around the eye, you can see those really cool white scales. And those are characteristic of the lycaenids. Now, if we're looking at the wings, I said maybe we would see more blue. So let's see if we can find a place on this guy that's still a little bit blue. So you can kind of see it. It looks like a faded blue. Almost like a pastel blue on the microscope. I promise it's a little stronger in real life. Let's see. Maybe if I play with the white balance. Not that way. Ha! I could make it so blue! No. Probably something like that. Awesome. So those are our lycaenids. Okay. Now, we got all those taken care of. The eyes, the antenna. The fact that they are itty bitty and delicate is a thing. I can tell you, it's very difficult to spread and pin small butterflies and moths. These big ones, those guys are easy because they're huge and it's hard to tear those wings. They're very, very strong. But when you're talking about the little itty bitty moths, it's so incredibly easy to rip a wing. And you really don't want to do that. Alright, so the last family of butterflies that we're going to be doing are the Nymphality. And these are called the BrushFooted butterflies. Alright, there are many brush-footed butterflies out there in the world. I'm not sure which family is the largest. I'd have to look in to see what family of lepidopterans is the largest. But the brush-footed butterflies, there are many of them, and one species is the monarch butterfly. So the monarch butterfly is a nymphalid. I can tell it's a monarch and not a viceroy because if I'm looking at these hind wings, all of these black veins, they go out towards the edge of the wing. All right. There is another butterfly called the viceroy that mimics the monarch butterfly. So it looks almost exactly identical, except that on the viceroy, there's another cross vein that crosses all of these veins. And it's black. So there's a black line that goes from here all the way up. And so that's going to be your characteristic you can look for if you're comparing monarchs and viceroys. All right. But all brush-footed butterflies have their front legs are reduced. All right. So if you see a butterfly and it looks like it's only standing on 4 legs, that's because you can't see its first 2 legs. Their first 2 legs can be kind of small and not super obvious. We're going to turn this monarch butterfly upside down so that you can see that. All right, so we are looking. All right, so this is the hind pair of legs right here. This is the middle pair of legs. And so we want to move up a little bit. Okay, so we see our head. I can see them. This might be a little bit more difficult to explain. All right, I'll just show you really quick. This is the middle leg, so if you look right here, this is going to be the femur, this is the tibia, and then the tarsi start here. And then we're going to be looking for another pair of legs that's a little further up. You see this, what looks like these two segments right here and then right here? That's going to be the greatly reduced front leg. All right, you'll notice that it is less than half the length of the middle leg or the hind leg. It's very, very tiny up there. I wonder if yeah, so that right there, right here, that's the greatly reduced front leg. A lot of times, maybe it would be a little easier to see it if I turned the specimen sideways. I wonder if this will work. We should try this. No. Me, myself, and I are going to have to hold it, I think. Alright, so I'm using both of my hands, so I can't use my mouse or cursor. But if you look right up there near the... If you look right up there near the head, you can see the compound eye. And then if you look down a little bit, there's that shiny kind of smaller line. That is the little brush-footed leg. Okay, so that's going to be the little itty-bitty... That's going to be the little itty-bitty front pair of legs that this whole family is characteristic for. Characteristic for the entire family, have those little brush-footed legs. Let's zoom in. Cool. Those are the scales. Alrighty, so those are the brush-footed butterflies. The monarch butterfly is an example. The Nymphality. The bloop. What's the other one? The viceroy is also a brush-footed butterfly. Alrighty, and so those are going to be the butterflies. The rest that we're going to go through are moths. Admittedly, I love moths. I think that they're gorgeous. And a lot of people say, but moths are kind of boring and brown and don't have color. I would strongly disagree. I would say that moths can be just as colorful or more so than butterflies. Okay? Just because moths are flying around at night doesn't... a lot of them are brown and boring, okay? I know. But there's a lot of beautiful ones out there that we also want to give credit to. Okay. I have to put the labels back on my monarch. Don't let me ever forget to not put my labels back on my specimen. That would be sad. I was thinking about that recently because I take the labels off to show you guys characteristics and for me to see characteristics easier. I need to make sure that every time I take these labels off, I put them back on. Don't want to lose any. Alright, we're going to be talking about these two next. These are part of the family. I believe it's Lazocampid. Give me 2 secs. Yep, there it is, Lazocampid. Alrighty. Lazoacampids are the tent caterpillars. So these guys, these are special tent caterpillars. We don't have these in Michigan. We have other species of tent caterpillars, right? Lazocampid is a huge family of moths, and they all, or most of them, make tents in trees. So they'll create a lot of silk with their mouth parts, and they'll weave leaves around. And depending on the species, they'll either they'll weave leaves into their home and then just eat what's in their home, or they'll live in the home and forage outside of the home. It just depends on what species you're looking at. Now, when we are looking... Oh, excuse me. It might be difficult to see. Some characteristics are really hard to see. I mean, I could convince you that it was there, I think. I'm gonna look these guys up really quick. Glovaria arizonensis. Alrighty, so these are the Glovaria. These are Lazoacampids. I remember having a hard time trying to get this guy down into family. It took me a very long time. Alright, I know that this is Glovaria arizonensis, right? So it's a Lazocampid from Arizona. They are actually pretty common out there. Now the front wing is, the characteristics are in the wing venation. Alright, so we're going to be zooming into the wing venation and trying to see that together. Some other characteristics that you'll notice, a lot of Lazoacampids are super, super, super hairy. Alright, like more setose than normal moths. Alright, so you'll notice that they have large tufts of hair all over the place. They're super, super fluffy. I had a previous bug collecting partner that used to call them the Chewbacca of the moth world. Alright, so that gives you an idea as to how fluffy they're going to be. And then their hind wing has a humeral angle. But that is going to be a little bit more difficult to see. So hind wing with strong angle, humeral angle. We'll look at that. We'll try to look at that. I'll try to point it out to you. We'll see if you can see it. They are super fluffy. Alright. And we're going to say... Alright. We're going to say that the front wing has a CU vein with 4 branches. And we'll show you that. We'll see if we can show you that. I think that we'll be able to see the wing venation, and you'll definitely notice how fluffy it is. The humeral angle, I'm not sure if we're going to be able to see very well. We're going to have to flip the specimen over. Right? See how fluffy? He's so fluffy, I'm gonna die! Does anyone know that reference? Okay. Oops. All right, here's a fun little mini factoid. I think our vein that I'm looking for is in that white spot, which is why I can't find it. I believe that is exactly where the vein is. It's gotta be it. All right, they are super, super fluffy. All right, so we are going to pretend. We're good at pretending. Here, I'm going to draw you a sketch really quick. This is what we are looking for so that you can kind of look for it with me. We're going to imagine we have a front wing, and then something like that, and then, you know, stuff that comes off that doesn't matter, and then 1, 2, 3, 4. Okay, so let me show you really quick. Now, if we're looking at the front wing, okay, we have, there we go. We're looking for essentially this box right here. And then we're going to be following this box, and we want to see how many times the bottom splits off, starting at like right about in the corner. There should, in theory, be 1 and 2 that meet here in the corner, and then another one that comes off a little further back, and then another one. For this family, we should have 4 that come off the front wing of our insect. Alright. Now, if we look at our microscope, I believe that the end of that rectangle is right there on that white spot. It's right here. Because we can see that there's a vein that comes off this way and a vein that comes off this way, right? And so if those guys are meeting right here, that means that there's likely another vein that goes up this way. Now, there's so much hair and so much scales on these wings that I can't see the vein. All right? So, this would be a specimen that I would have to wipe the scales off to see that. I'm not going to do that today. So we've got 1, 2, and then if we wanted to count 3 and 4, you see right here, that's going to be a darkening, that's a vein. So 3 branches off from this. And then if we go a little further back, I'm sure that this branches out, but you can see it right here. This is branch number 4. All right, so it goes 1, 2, 3, 4, and then it comes up and goes back. All right, so that's going to be characteristic for this family, that the CU vein, or these veins that are on the bottom side of the square, breaks off into 4. 1, 2, 3, 4. All right, the other thing that we can see from the top, oh yeah, is that it's hairy. All right, let's flip it over. We want to be looking at, they call it the humeral angle, or it's essentially the inside of the hind wing where the hind wing meets the forewing. This is as far out as I can get. Yeah, this is going to be hard for you to see. He's too fluffy! Alright, so, this is the front wing up here. This is the hind wing down here. So you can see right about here, this is where the front wing and the hind wing separate. Okay, so this is the edge of the hind wing. It's going to be really hard to see. But it has what we call a strong humeral angle. So right here where this hind wing ends and then it goes to connect, there's this strong angle right here that is nearly impossible to see on this specimen. Let's see on the other side if it's any easier. Well, at least you can see the front versus the hind wing easier. The line is right here. But then it kind of comes up and comes down, and the hind wing connects here. So you can see that this angle has to be kind of large. I'm not sure what the purpose of that angle is, outside of maybe it holds the hind wing behind the front wing so that they don't get all flipped around. I don't know. That would just be a personal guess. Cool. Alright, so those are the Lazocampid. So now you know, when you see those tents outside on the trees, when they emerge, they're gonna be super fluffy moths. Right? Okay, so we only have, let's see, we have 1, 2, 3, 4. We have 4 families left to talk about, so it's probably gonna go about another 20-ish minutes. The next one that we're talking about are the Saturniides, or the silk moths. Now, the Saturniides tend to be the large moths. 90% of the time. The Saturniides are very, very large. Now, that's going to be a characteristic that you can't use 100% of the time, right? Because that is arguable. And the idea of large changes from person to person. But they do go from large to very large. Or XL. They can be super big. They will be the largest night-flying moths that we find here in the United States, I believe. Let's see. Outside of the black witch moth. Let's see. What family is that in? The black witch moth is an erebid. So that is it's closely related to noctuids. Anyway, I needed a little bit of learning to myself time. All right, so these 2 big ones right here in the front that you guys have all been staring at that are beautiful, those are our Saturniides, or our silk moths, that we're going to be looking at today. This one on the top is an imperial moth. And this one on the bottom is what we call a polyphemus moth. Alright, a lot of times with these large Saturniides, the way that you tell the males from the females is that the males have large plumose antennae, and the females, their plumes on their antenna tend to be a lot smaller. Alright, now the actual characteristic for this family is a wing venation on the hind wing. Oh, cool. We can see it from the bottom. I was going to say, this is going to be a characteristic that's nearly impossible to see from the... It doesn't fit. Yeah, so this characteristic is going to be nearly impossible to see from the top, but we'll be able to see it from the bottom. So that's our silk moth close up. Cool. This is an imperial moth. This one is a female. Let's see. collected this year on my collecting trip in Arizona. It might not be an imperial moth, huh? I need to look that up, if there are imperials in Arizona. Ooh. Gentle with their wings. Okay. Let's get this moth flipped over so we can ID it. Look at his cute little face. Oh no. Don't do that. Oh crap. Well, that's no good. Can't do that. Give me a moment while I try and figure out how to flip this guy over without hurting him. There we go. So I can't use museum tack with moths because the museum tack pulls all of the scales off of their body. He's got a bare spot on his back now. Whoops. Alrighty, so what we are looking at right here is the hind wing. Okay, so this right here is the edge of the hind wing, and up further, this would be the front wing. This is the first. This is the first vein that you can see on the hind wing, and this one right here is going to be the second one. It goes right about here. It follows through. Let's see. And then they run about parallel to each other. Okay? But our characteristic for this family is that this is the SC vein. The SC vein diverts from the R vein. It just means that this has an outward arch, or it arches away from the other vein. I would have to look at other specimens of butterflies and moths to see what kind of the normal is. My guess is that normally it would run more straight or more parallel with this vein in the start. Some silk moths have a very drastic angle right here. Some of them can really push far away from the R vein. This one is a little bit more subtle, but that is the characteristic for the family. Let's see. Oh, and if you want, you can see. I'll have to look at before and after pictures, but you see how he has a little bald spot underneath the pin? Poor guy had got some museum tack stuck to him. Hi, TJ. I am doing well. I'm glad you came to hang out with us. Do you have a favorite one yet so far? I think that they're awesome. Alright, so those are Saturniids. Let's see. I've got a Luna Moth over here. I figure we can kind of show you the Luna Moth, and the Luna Moth is also a Saturniid, so it's also going to be one of those moths that you can say are kind of those large night-flying moths. Here, let's check him out real quick. Alrighty, so that is going to be our Luna Moth. Our Luna Moth is going to be essentially even longer than the other two specimens. It's why I kept it in a separate container. Cool. I love these Luna Moths. A lot of times these big silk moths are going to be flying around most commonly in the first really hot night of the summer. So if it's still like 80 degrees at nighttime and it's the first night that it's done that, that's when the big silk moths are going to be flying most of the time. If we take this Luna Moth and we flip it upside down and throw it under the microscope, or very, very carefully place it under the microscope, we can, instead of taking this label off, I think we can just turn it. There we go. Now, we were talking about that SC vein and the R vein being that first and the second vein coming off. This is the SC vein. This is the SC vein right here, and you can see that it goes up and diverges, right, from the R vein. And that's going to be the significant characteristic, is that this vein kind of pushes further away from the R vein, and it follows up. Very cool. All right, so those guys are the Luna Moths. Let me see. I don't think I have any more. The Polyphemus Imperial. I think they're good. So we might as well look at the Polyphemus antenna, because I promised you guys at the beginning of the live stream that I would. Now, the Polyphemus antenna, this is a male. So he has the huge feather-like antenna that you'll hear about on moths. Check that out, right? So these silk moths have the ability to, does the amount it diverges depend on the wing shape? The amount that it diverges can depend on the wing shape, but a lot of times it actually depends on, let's see. Yeah, I guess. It just depends on the species, really. The wing shape is going to change the angle of the SC vein as it kind of follows the front of the hind wing. It's going to be the leading and strongest vein down there, so it's going to be kind of what helps hold structural stability on the hind wing. Now, that, because it follows the wing, yes. But most Saturniids are going to have very similar hind wing shape. So most of the Saturniids or the silk moths are going to have it's going to be a very similar shape, even though they'll be kind of different sizes. All right, the males have these really awesome feathery antennae, and they have the ability to smell the ladies from incredible distances away, sometimes even miles. All right, so yeah, the one I'm holding right now, we're checking out under the microscope. This is called a Polyphemus moth. It's native to well, the eastern United States, I guess. I found them in Michigan and in Pennsylvania. I don't know what its natural I don't know how far it heads out west. But, yeah. And I thought that we can check those out even closer. Cool. Yeah, so that's what his antennae are going to look like. And while we were looking at the Polyphemus under the USB camera, I noticed that the blue scales on this eye spot are very, very drastic. So I kind of wanted to zoom in on these scales and see if, yes, that's so cool. Guys, check that out. These are the scales on the butterfly's wing. Alright, so these guys are going to be the little itty bitty scales. This is where our moth gets all of its coloration from. I like that the scales on this one's wings are kind of like forked at the end. A lot of butterflies and moths, they have scales on their wings, but they'll have kind of like different shapes. Different shaped scales. I believe that this spot right here that you see that almost looks like a piece of plastic, I believe that's see-through. Yeah. That is a clear spot in the wing that you can see all the way through. And so that's why if you go to hold a butterfly or a moth, you end up with that like powder or that kind of fluff all over your hands. That powder is actually the scales that have rubbed off on your hands. And if you rub off enough scales from a butterfly or a moth's wings, you'll actually, you can actually make them completely clear. Alrighty. Activate. So we're going to write down that characteristic for the hindwing vein. Saturniids are large to extra large moths, but they also have an SC vein that diverges from the R vein on the hind wing. And we saw that. Cool. All right, the next one we're going to do are the Sphinx moths. Sphinx moths. I have more Sphinx moths than any other family. I really enjoy collecting them. They come to my blacklight all of the time, and they fly between like 11 p.m. and 1 in the morning, and so they're in the perfect, window for collecting when you're out and about and having a good time. One of the things I love about them. Now, the defining characteristic, the one that you'll see 90% of the time, is that the hind wings are going to be about half the size of the front wings. All right, so if you're looking at that, the hind wings are going to be half the length. Let's see. Let's get a cool one. Let's get this one. This one's pretty. That one's pretty too. There's so many beautiful Sphinx moths in the world. All right. This one's pretty. All right, so if you are looking at this Sphinx moth right here, I believe this is a white-lined Sphinx moth. And you can see back here, we've got the hind wing, and up here we've got the front wing. And if you're comparing the size, hind wing, half the size of the forewing. That's going to be a pretty easy characteristic for you to see. And then the other characteristic is also the shape of the wings. So the front wing is super narrow. So these guys, the Sphinx moths, can be identified to family easily without looking at the wing venation or flipping them over by just looking at them as they are. All right. I believe that this guy was actually a Guess That Bug one of the weeks ago. Let's check out some more. Let's see. I have so many. I love, this is the one that I actually picked out to show you guys because it has these beautiful pink hind wings. And a lot of times with Sphinx moths, oh, look how pretty. A lot of times with Sphinx moths, they can be pretty dark, and this overall has a super light coloration to it, so that kind of makes me happy. It's more pastel. So that is going to be Sphinx moth. Let's see. Other Sphinx moths, they can have a variety of sizes. All right, this one's awesome, but it needs a container without walls. All right, Sphinx moths can even be ginormous. All right, but you will still notice that the hind wings are about half of the size of the front wings, even though this guy is about the size of I don't know, about the size of my hand. This species of moth actually comes in two color forms. This is the light color form. I did not collect any of the dark color form when I was collecting these guys. So next time I'll find them. The other moth that's a sphinx moth that people know really well, sometimes they call it the hummingbird moth, looks like this. We can actually, I think this guy would be really cool to look at under the microscope because he's a little bit smaller. So it'll be a lot more manageable under the microscope. But also, because the wings on this moth are see-through, most of the wings don't have scales. Now, Lepidoptera, right? That's the Latin name for butterflies. Lepi means scales, and Ptera, ptera means wings, like a pterodactyl, right? The winged dinosaur. Now, these guys their characteristic is that they have scales on their wings. All Lepidopteran do. Now, this one doesn't have many scales on its wings, but it still has them. He's like, I'm still a moth, I promise. Or a butterfly. Nope, moth. He's a sphinx moth, right? Okay. So, if we look at where the color is on the very tip of his wings and we zoom in, that's where we're going to see these scales. So you can see all of these little rectangular scales. This is where he's getting the color from on his wings. And then, if you look further into this area where it's kind of just open and white, that is completely clear because he's got no scales in that section of his wings. This definitely was a Guess That Bug recently. Alrighty, so this is a snowberry clearwing. This isn't the hummingbird moth, but it's in the same genus. So a lot of times people would refer to it as the hummingbird moth anyway. Okay. So, sphinx moths. That's the six-spotted sphinx. This one is really cool because he has the ability to camouflage really, really, really well into his environment. He literally looks like bark when he lands. Alright, so those are the sphinx moths. The next one we're going to talk about are the Noctuidae. Now, if I could only remember the common name for the entire Noctuids, these are going to be, I mean, people call the family prominence. Or oakworms. But those aren't really common. They are the common name, right? Those are going to be the common names for the entire family, except that they are not commonly used by people, all right? Because there's such a huge variety of noctuids out there that people either call them noctuid moths or they call them by a more specific name. For instance, this is a type of noctuid, and we would call it an underwing. Alright, you can see that the front wings are those super well camouflaged kind of mottled coloration that hide really, really well on a tree. And then on the underside, they've got those, that striping and those colorations. Now, this is common for underwings, right? And there's a variety of different colors of underwings, from red to orange and yellow. Let's see. There. All right, these are all noctuids. These are all underwings. And you'll notice that they all have that same type of striped patterning, but they can have a variety of colors. Colors are really difficult to identify moths too. Colors are just really difficult to identify things by because they can fade and change over time. A lot of times we're going to have to look at the wing venations. So, if we are looking at the wing venation, let's write down some characteristics to look at. So, the first thing that we're going to talk about are the median veins, or the M veins. And we're going to just, I'll tell you, and then we'll show you. All right? The M2 vein is going to split the discal cell, the D cell, and not the battery. All right, the discal cell in the wing. If we were looking at the front wing, for instance, of this monarch butterfly, the discal cell would be this one, right? This large closed cell. That's the D cell. All right. Do our guys have those? Kind of. Wow, they're so pretty. I'm gonna have to show you one of this one is like metallic in places. All right, so really the characteristic that we're gonna be looking at that's solid is this M2 vein splitting the D cell. The other ones, we'll look for. At the inside edge of the front wing, it has backward projecting tufts of hair. All right, so it has... All right, so it has these hair tufts. If you imagine its wings are up and on the inside, or essentially on the armpit of the butterfly pointing backwards, they're going to have long tufts of hair. So we can check both of those characteristics out under the microscope. All right, backwards facing hairs is easy enough. These guys right here, these tufts of hair is pointing backwards. It's just that I know that there's a number of moths that have this characteristic. So this is just kind of one of a couple characteristics. You can't say, oh, it has all of this hair pointing backwards. It's a noctuid, right? You're going to have to look for another characteristic, but it is something that you can use. Now, we're going to see if we can find the discal cell amongst all of these wing hairs. Ugh. Wing scales. Let's see. Discal cell. Ooh, baby, 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 discal cell. Doo-doo, boop, boop. Oh, no. Oh, no. Oh, no, 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 no. I think it's right there. All right, that's going to be impossible on that specimen. We're going to check this one. sure about this one. Well, this is interesting. We're trying to find the D cell on these moths' wings, and honestly, they are very, very difficult to find. You kind of have to look for patterns in the wings. Now, one of my favorite things was Dr. Bird, one of my professors in university. He once said to me, if you can imagine it, it's there. And I know that's not super scientific, but sometimes I'm looking through a microscope and I'm thinking, I don't think it's there. And then I think, well, maybe if I imagine it, I'll be able to find it, and then I'll have, something to go off of. All right, so let's just go with me here. Now, this line, you can almost see pretty clearly, right? This is a vein right here. So it's kind of dark. It has this lighter side on top, and it moves through, okay? And I think that it ends right about here. Now, we also have a vein that starts here, and it moves up. I lose it in this general area, but I think that it goes something like this. Alrighty. And so we've got that, we've got this guy, and then, right about here maybe, we have a cross vein, and that would give us a discal cell. Alright, so I'm going to say that that's our discal cell right here. And now if we're counting the medial cells, we're going to be counting, there's M1, M2, and M3. I lied. I'm going to call these noctuid moths because I've been calling them that anyway. So these are noctuid moths, and I'm just having a really hard time with this discal cell. All right, so here's the issue. That is going to be a key. That's going to be a key issue with identifying butterflies and moths a lot of times, is trying to figure out oh no, I think I froze. Did I freeze? I swear I'm here. All right, we're going to take that off until I unfreeze. Huh. Okay, so that's an issue with identifying butterflies and moths, and it's that a lot of times when you're looking at them, we kind of warned you earlier, these guys are going to be very, very difficult to identify. These guys are going to be very, very difficult to identify without knocking the scales off of their wings. But you can see that they have common characteristics, right? They're all noctuids. Now, they do have those hair tufts, but the other characteristic, I found one that we'll be able to see really easily that's not in the wing venation, and that's that they have ocelli. And normally they're at the top edge of the compound eye. Alrighty, so if we flip over and we check out our microscope really quick, and let's see. love characteristics that are impossible to see because the guys are fluffy. There's the compound eye of our moth, though. Alright, I have to restart my face camera, so give me 2 seconds. Well, that might just be a problem that we live with, ladies and gents. Alright, so those are going to be our noctuids. I was having a hard time finding that ocelli too. So, I'm sorry we couldn't see those characteristics, but I promise that they exist. And if we had, I don't know, if I wanted, I probably could knock off those scales and we could dig for them. I don't really want to look into doing that right now. All right, the last family we're going to be talking about are the arctiids. These are generally known as the tiger moths. They tend to be very, very pretty, except, well, let's change that. They tend to have very, very vibrant colors on their hind wings, and on their body. A lot of times their front wings are pretty dull or black and white striped, and that's why when you look at them, you kind of want to scare them, and you want their wings to open so you can see how pretty they are. Those, we've got a couple of them. Let me take these underwings off of our tray really quick. Okay. Now, our arctiids, or our tiger moths, let's see, I think these are the two best ones. They look like this. They tend to be small to medium-sized moths. These are a little bit faded, alright, so you can see that they have this guy, he's got these black and white stripes up here on the top, and then the hind wing is actually a dulled pink color. It used to be a very vibrant pink. It's dulled over time. And the same with this guy. He's got those dull kind of brown front wings, but then the hind wings, you'll notice that they're kind of pink. Pinks and blues are fans. Tiger moths like to be pinks and blue colors, which is great because I love when moths are pink and blue. Now, their abdomens have these cool, pretty, their abdomens tend to be very brightly colored. They also tend to have, let's see, like stripes of color and then segments of black spots. That's a pretty common coloration on the abdomen of arctiids. All right, let's look at the wing venation. We're gonna look at the wing venation and we're gonna see if the characteristics I want to tell you are there. Hey guys, guess what? I'm back! Awesome. That only took a minute to fix. Okay, let's check out these tiger moths. I think that we'll be able to see this. Maybe not. I see 3. See, the funny thing is, I'm supposed to see 4 veins. And unfortunately, I'm having a hard time imagining this one into existence. Alright, this image of the microscope should give you the characteristic that we are looking for. We are looking for the, for this vein down here to be branched 4 times. So, this is the vein we're looking at. This is the first branch right here. I think that that one is at least pretty obvious. Alright, so that's branch number 1. It goes a little further, and then I believe this right here is branch number 2, and this is branch number 3. You can see this like here. Maybe it comes like that. Except that this family is supposed to have 4 branches. There's supposed to be another one up here. And I'm not seeing the 4th one. Let's check the other specimen. Aha! This one is super obvious. Okay. I like it. This one, this is the D cell right here. It goes all the way around. It's large and it's enclosed. All right. Then you have down here the first branch, branch number 1. Then you have branch numbers 2, number 3, and number 4. And that is going to be characteristic for this family. It's that the CU vein appears to be branched 4 times. 1, 2, 3, 4. Front wing. But you can also see that they can also just kind of be beautiful. They when they're freshly caught, these colors are dynamic. They are incredibly bright, especially with this pink coloration. You almost be startled when they open their wings and you're like, "Wow, I thought you were boring. As it turns out, you're beautiful." Right? And the same with this guy. I don't know. I do kind of love the black and white striping on the front wing. So I think that that's very unique. But then when they open it up and it's pink on the underside, that's just awesome. It's like the moths are trying to show off to the world. They're trying to say, "Hey, hi, Space Cats. How are you doing?" Oh, that's cool, Space Cats. That's fun. Yeah, I've been working on an insect collection on the live stream, so I think this is actually the last order that we're going through and identifying things to family. And then I was actually trying to figure out what we are going to be working on next. And so something that I was considering, I've got 2 thoughts. One was working on identifying some of the families down into species. So going through and keying things out with people and getting really into the characters. Or I do have some backlogged insects that I still need to spread and pin. So I thought that we could pin and spread insects together too, and just kind of hang out and talk about bugs while we were spreading and pinning. What do you guys think? What sounds more interesting? Both are good? That's awesome. So I'll be working on that, and we'll be coming back on Thursday. I'm closing in. That was the last family that, right? It is really hard to choose because I think that both things hold a lot of merit, right? Maybe I do both and just split them. Say Mondays I pin and Thursdays I ID. I could do something like that. Was there anything that you guys missed that you wanted to see? I was trying to see if there were any other butterflies or moths that would be interesting. This one. I don't know. Right? Well, I hope that everyone is having... I love that you guys rated me and you're now here, except we were just finishing. So now I feel like we should hang out and do another thing or two. But I don't know what we would do. Do you guys want to look at some aquatic insects that I collected recently under the microscope? Think that could be fun? All right, let's do that. I have to go and grab a petri dish. I'll use this. Luckily, I've got all my bug stuff right here. Ugh, okay. There we go. Now, I have some buggies. Now, there are just a couple in here, but I figured we could chat about them. Wrong way. Readjust the microscope a little bit. Closer specimens. Now, I am not sure how much you guys know about aquatic insects. Alrighty, but this guy right here, let's get myself a pin. This guy right here is a mayfly. All right, he's in the order Ephemeroptera. And when we look at our mayfly, a lot of times the way that you're going to be able to identify these down to, at least down to order, is that they have these three tails. So right here, those kind of three long filaments, that's going to be how we identify these guys to at least order. This guy is a really, really fun family. I'm going to forget the name right about now, but he is an incredibly flat body. And if you think about water running over a rock, right? Here in a river, and the water is going really, really fast over top of that rock. Now, this insect, the mayfly, has to hold onto the rock with all of its might. It has to be super, super strong to be able to not be blown away by the current, right? Except there's this really cool thing that happens with mayflies. Mayflies are so thin that they actually fit within the, essentially like the friction line of the water. So if you imagine a rock and the current going over it, the closer and closer that you get to the rock, there's actually a very, very small area between the rock and like the fast current where the current is not moving very quickly, all right? Just because of the friction between the water and the rock. All right, so there's that space where there isn't as strong of current, and that's where these guys like to hang out. They stick, and they're so flat against the rock that the current doesn't affect them. Now, if we were identifying this guy down to family, we would have to be looking at, a lot of times we would be looking at the gills. So, my mayfly does have to breathe underwater. Alright, let's see. If we look at at his sides, these right here that look almost like large plates, those are the gills. That's how he breathes. Alright, and you can even see what kind of looks like veins inside of those gills. Alright, so that's going to be on my mayfly with 3 tails. I have another aquatic insect with 3 tails, and that is a damselfly. Alright, this is a damselfly nymph. So, very closely related to dragonflies, right? Except that they do have external gills. So a damselfly's gills are actually back here. Those 3 appendages right there, those what look like tails, those are its gills. Cool. Other than the nematodes. Oh man. Well, Dr. Bird, I mentioned that I had a professor say that if you imagine it, it's there. That was when I was studying and working with nematodes, because they're so incredibly small. It's like, Dr. Bird, I don't see the character we're supposed to be looking at. And he's like, if you zoom back and forth and then imagine that it's there, you can almost see it. Oh. Alright, this guy right here is kind of fun. You don't see them really, really regularly. It totally is like that. Yeah. Alright, this guy is a rock. No, he's not a rock, he's an insect. This is what we call a water penny. Alright, it is a water penny is actually an aquatic beetle. I believe it's a beetle larvae or grub. And so it looks from the top like it's nothing, right? It almost looks like it could be a rock scale, right? But they do walk around on the rock, and they have and they do have legs. So if we flip this guy over, and maybe we'll zoom in. Alright, so we can see way up here, this is our head, our antenna, and 1, 2, 3 legs, right? So our water penny has all 6 of its legs and its head. He walks around and has a good time in the water, I'm sure of it. I collected a couple of them because I think water pennies are super cool, but also I hadn't seen them in a while. And so I wanted to have some specimens to show off to people. Oh, this guy's big. Look at this one. Whoa. I hadn't looked at these guys under a microscope yet. They were sitting here waiting for me. It does look a little bit like a horseshoe crab. Itty bitty tiny one. And they do have those very similar gills too back there. Now this guy, he's a broad-shouldered blah blah blah. I believe he's a mesoveliid. Let me look that up real quick. Nope, he's not a mesoveliid. He's a... Give me 2 seconds while I work on my cheat sheet. I know that the characteristic for this guy is that he has this very characteristic shape, and he has these very broad shoulders. And he's called a broad-shouldered something. He's called a broad-shouldered something or another. Let's see. He is a hemipteran, or a true bug, meaning that it does have a piercing and sucking mouth part. I know that about him. All true bugs, you guys may not know this, because you're coming from fungus people. All bugs are not insects. Or, all insects are not bugs. Alright? Kind of like all squares are rectangles, but not all rectangles are squares. You can also say all bugs are insects, but not all insects are bugs. Alright? A true bug is a category of insects. Alright, so a true bug is a type of insect with a piercing and sucking mouth part. Man. Wish I remembered his name. We're gonna look it up a different way. You know what? I know where I can find it. Ladies and gentlemen, sometimes when the internet fails you, you go back to a book. I've got a book and I know where I can find this answer. So these are my notes from my aquatics class. From a little bit ago, you know? But I will admit, it's been super, super helpful that I took really complex notes back when I was doing this. Let's see. I thought I'd be able to find him. You know what? He might just be a mystery bug until I... Aha. A Veliid. A broad-shouldered water strider. We win. All right. It's not a mesoveliid. He's just a veliid. That's what messed me up. So, if you... Let's see. I graduated in 2013, so it was almost a decade ago. So this is going to be our veliid, or a broad-shouldered water bug. And I think, yeah, that's gonna be what I collected from that trip. I I hadn't collected a lot of aquatics, but I was just kind of picking around for a couple that I needed for my specimens for my collection. So I want to say thank you guys for hanging out. Space Cats, I'm going to have to come and see one of your live streams too. When do you live stream? While I'm putting this stuff away. All right, cool. So I just wanted to say thank you for hanging out. I had a really good time identifying butterflies and moths. Had a good time pulling out a handful of aquatic specimens to take a look at those. And we will be coming back with identification and more pinning and spreading. Thursdays we do it earlier. Thursdays we go from like 2 to 3 or until we go. Thursday from like 2 to 4. And then Mondays we start at 9 and, you know, we go until we end. Perfect. Thank you, TJ. I'll have to go see. Alrighty, ladies and gentlemen, I hope you guys have a wonderful rest of your day, and I will see you on Thursday, unless I see you at Space Cats live stream on Wednesday. Alright, bye!